The eLife Podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, welcome to episode 51 of the eLife Podcast. I'm Chris Smith from The Naked Scientists. In this episode, the effect of climate change on the spread of mosquito-borne diseases, lightning strikes twice as another transmissible cancer surfaces amongst Tasmanian devils, and new insights into how LSD works in the brain. But first, one prediction is that by about mid-century, over 3/4 of the human population, and that, by the way, is destined to reach 10 billion by then, will live in a city. And cities themselves will, of course, need to grow to accommodate this burgeoning populace. But where they'll expand into will inevitably be the habitats of wild animals. With what effect? Ariel Parsons has been trying to answer this question in the US.
The idea is that these cities are out of ecological balance, that humans have taken away too much of the natural habitat and too much of kind of the natural systems. And so they're not really functioning to the same level as we would expect of wild ecosystems. And so we wanted to look at that in terms of the mammal communities and compare two cities in the United States, Raleigh, North Carolina, and Washington, D.C., to nearby wild areas and see if we could see any differences in terms of diversity, so the number of animals present, and then the relative abundance, so kind of the population sizes or some estimate of the population sizes of each species.
That sounds like a very tall order. How did you do it?
It is indeed a tall order, and so we had to cover these really large areas in both cities to get kind of an idea of these communities in the very suburban areas all the way out to the more wild areas. And so to do that, we used citizen scientists to run camera traps, devices that we can put out in the forest or in a yard, and when an animal walks by, it senses the body heat and the movement, and it automatically takes a series of pictures. Without these volunteer citizen scientists, we simply couldn't have covered the areas that were necessary for this study.
Where were they deploying their cameras, though?
We asked them to deploy them in different areas around these cities that were based on housing density. So areas of highest housing density are what we call suburban areas. And then as we step down in housing density, we get to these wild areas. Citizen scientists then put those cameras in yards and also in forested areas, both small forest fragments, less than a square kilometer, and large forest fragments.
And how do you log all the data?
We use a software called eMammal, a network that helps camera trappers organize their data. And that software allows our volunteers to go through all the pictures that they collect and identify the species in them. Then they upload the data to us so that we can look at it and make sure that they've correctly identified species. There are some tricky ones here.
Why?
Why are they tricky?
Yeah,
so we have two fox species. We have the red fox, and we also have the gray fox. And they're of similar size, and especially at night, these cameras, they're black and white. It's an infrared flash, so they can't see whether the coat is red or black. And so there are some characteristics, especially of the tail, that make it easy to distinguish, but our volunteers sometimes have trouble.
But assuming that your volunteers were diligent and they weren't too easily confused, what did you find?
So we found, rather surprisingly, that the diversity, so the number of species that we were getting on our cameras, was not significantly different between the suburban areas and the wild areas. And in fact, in Raleigh, the diversity was significantly higher in the suburban areas compared to the wild areas. We did have some exceptions. For example, we only found beaver in the suburban areas, and we never actually detected it in the wild areas. And we never got bears in the suburban areas.
I guess that's a bit of a relief in some respects, isn't it?
Yes, I think it is. At the same time, we know that bears actually can do quite well in suburban areas. The tricky thing is making sure they stay wild, so especially not letting them get into trash cans and allowing them to maintain their distance even if they're living close to us.
Does one take away from this then that, contrary to the prevailing view that these urban environments are really bad for wildlife, in fact, the opposite's true?
Yeah, Yeah, I think in terms of these mammals, and it is important to say that these mammals that we were sampling with our cameras are mammals that are chipmunk-sized and up. So there are a number of mammal species that this wouldn't apply to. But certainly in these two cities, it seems as if this idea of ecological function being different between suburban and wild areas, at least as it applies to the mammalian community, is maybe not the case. But there have been a number of studies on other taxa showing that urban areas and suburban areas do have quite drastic negative effects. And so we want to be very cautious about extending this, to other taxa and other cities even.
So what about other cities? Because obviously you've looked at just two. You looked at a lot of animals in a big area, admittedly, but it is just two zones, isn't it? So what would happen, do you think, if you went elsewhere, both in the US and maybe even further worldwide?
Yeah, and that's something that we're very, very interested in doing. We're really interested in looking at cities that have an impact apex predator. In the East, in these cities, of course, there were once mountain lions and wolves. They are here no longer, but there are cities in the United States where the foxes, the raccoons, etc., still persist in the presence of an apex predator like a mountain lion or a wolf. And we would expect then perhaps the dynamics to be a little bit different. And then the other thing, kind of thinking about cities around the world, of course, cities in different parts of the world, in Europe, for example, tend to be more concentrated, less sprawling. The separation between urban and suburban and wild tends to be very stark, whereas here there's quite a lot of sprawl. And so we would then expect it to be more difficult for animals to go in those small, concentrated European cities from the wild straight into the suburban areas. But it's something that we're very interested in and looking into further. And we actually have some partners in Germany right now that are just beginning a replication of this study.
Oriel Parsons, she's based at North Carolina State University. Now, speaking of urban dwelling, most of the world's big cities are sited in places where the conditions are ideal for human habitation, and often our ancestors chose those locations for their settlements to avoid diseases. For instance, they didn't go near swamps, so they wouldn't be plagued by mosquitoes. But with climate change, in the future, some of these urban venues are going to become much more propitious for mosquitoes and therefore the diseases that they vector. So is there a way to tell which ones? Marta Schockett hit upon the idea of using Australia as a giant petri dish to find out. The country is so big that it exhibits a broad temperature range across the continent, and the use of a single healthcare system means there's a really excellent data set on at least one mosquito-borne disease, and that's Ross River virus. So she's been able to model accurately how temperature affects mosquito behaviour and disease transmission.
My lab was really interested in trying to figure out how temperature affects the spread of infectious diseases, and specifically infectious diseases that are spread by mosquitoes. We know that climate change is going to be changing the temperatures that we observe, and not just what those temperatures are, but when they occur in the year. And we know that temperature has a really big impact on the way that mosquito bodies work.
So basically what you're saying is if climate change changes the temperature changes the behaviour of potential vectors like mosquitoes, and that means that the dynamics of how diseases may spread will be secondarily affected.
Yes, exactly. And it's not even just behavior in the way that we typically think about it, like where they go and how far they fly, but every aspect of how their body functions, how long they live, how many eggs they lay, how quickly they turn from eggs into adults, how good their body is at transmitting the virus, because the disease has to get from their stomach to their salivary glands. And so that process depends really sensitively on temperature.
So given that, how can you make predictions about what impacts climate change is likely to have on things like emerging infectious diseases and the distribution of the diseases we already do have?
One way that you can do that is by building a model. And so by model, we basically just mean an equation that lets us turn body functions, things like lifespan, egg production, how good you are at transmitting the disease, into an overall prediction for disease spread. And so we can take lab experiment data where people actually take mosquitoes and put them in incubators at different temperatures and measure how all of those processes are affected by temperature. And then we can come up with an overall equation for how we think temperature affects the spread of disease.
That tells you what your model thinks is going to happen, but how do you validate it?
So that's a great question. So in order to test this model, we basically need to take human cases for different diseases and say, how much do these current cases of diseases match what we would predict based just on the model?
So what disease did you actually look at?
So for this study, we were looking at a disease called Ross River virus. It's the most common mosquito-borne disease in Australia. The reason why we were so interested in Ross River virus is that Australia is basically perfect to study how temperature affects mosquito-borne disease, because Australia is a huge continent that stretches from very tropical areas in the north to much cooler areas in the south. But throughout that entire range, there's a single case reporting system. So we have a really good data set for both the human cases, weather data, and in some cases, even mosquito data. We can use all of those pieces to try and test our model that was based just on data from lab experiments.
Right, so because you've got this broad range of different temperatures across the continent, and you can see how in the present situation that temperature and the seasonal data alters the behaviour of the mosquitoes, you can then use that to say, well, if climate change causes areas to become warmer or colder accordingly, we would anticipate that we'll see what currently happens in that geography happening in this new geography, and therefore this is what the likely outcomes will be.
Exactly. And actually, one of our main conclusions is that we expect to see different impacts of temperature increases in the different regions. So it turns out that there's a best temperature for the spread of mosquito-borne diseases, and it tends to be kind of a middle intermediate temperature. So areas that are already really warm, maybe like right at the best temperature, when they get warmer, we'll actually see that disease transmission will probably decrease because it will become too warm. Unfortunately, most people in Australia currently live in the southern parts of the country, which is right now below that best temperature. And so, in general, we probably will expect to see more cases of Ross River virus due to climate change.
So perhaps not the news you'd hope for if you live in Australia. Stanford University's Marta Schockett. Now, sticking with Australia, in recent years we've realised that some cancers can spread, not just around the body, but between individuals. And a very striking example of this is devil facial tumour disease that's wiping out the Tasmanian devil. We think that when these animals fight, cancerous tissue from one can be injected into another animal by a bite. There, it takes root and grows, using an immune trick to prevent it from being rejected by its new host. Recently, though, scientists discovered a new form of devil facial tumour disease, DFT2, which is distinct from the first one. This hasn't evolved to hide from the immune system yet, but if it does evolve to do that, then it could be curtains for the species. Hannah Siddle is studying these diseases at the University of Southampton.
Not many people will know that some cancers can actually become transmissible. And what we've been interested in is two transmissible tumours that have occurred in the Tasmanian devil, which is a marsupial species endemic to the island of Tasmania. And in the mid-1990s, a really nasty transmissible tumour emerged called devil facial tumour 1, or DFT1. And it's had a really negative impact on the population. So in some areas, 90% of the devils have actually died. But our most recent work is actually on a more recently emerged contagious cancer in the Tasmanian devil. So this is a species where lightning has kind of struck twice, if you like.
How does the cancer spread from one Tasmanian devil to another?
We think that this actually occurs by biting. Now, this new transmissible tumour, DFT2, which emerged only quite recently, also looks a lot like DFT1. And actually, if you looked at these two tumours, even though they're completely different diseases, they look really similar. So they still cause these huge tumours around the faces of the animals. So with DFT2, we haven't shown this experimentally, but we think that they're also passed by biting.
It was a number of years ago that Anne-Marie Pierce put some cells from the first of these types of tumours under a microscope, and she showed that they had to be clonal. It was exactly the same genetics in each of them. So how did this second type of tumour emerge, and how do you know that that's history repeating itself, or as you put it, lightning striking twice?
Yeah, that's a really good question, because actually, I mean, what you'd kind of assume is that this new tumour would be somehow related to the first tumour, right? That would feel more obvious, I think, because of how rare these are. But actually, when people first discovered this second tumour, and it looked exactly the same, but they found that it expressed some proteins on the cell surface of the tumour cells that were different to DFT1. And then looking a little bit deeper at the genetics, they found that actually the karyotype or the chromosomes of this new tumour were really different to DFT1, the first tumour. And then going even deeper and looking at some of the specific genetic markers, they were different as well. And they were also different to the host animals. So from this, it's really clear that this new tumour actually emerged in a completely independent animal, so a completely different animal that came from a different part of Tasmania and lived much more recently.
One of the first things that you published on this was the discovery as to why, when you put foreign tissue from one of these animals into another animal, the immune system doesn't recognise it and get rid of it. Why is that?
Yeah, so initially when we were looking at this disease in Tasmanian devils, we thought that it was just because they had low genetic diversity. And that's why we could have these cells grafting, if you like, as a graft between them. Because we know in humans and mice that you can't just take skin or a kidney and transplant it to someone else. It just doesn't work. The immune system sees it, it rejects it. And what we were able to show with the first tumour is that the tumour cells have downregulated or lost really important molecules from the cell surface, which are called MHC, class 1 molecules. And the tumour has cleverly lost these to make it invisible to a certain subset of immune cells called T cells. And so that's how DFT1 really probably became so dangerous and managed to kill so many devils.
It flies under the immune radar.
Yeah, that's right, yes.
Does the second type that you're now describing pull the same stunt?
Yeah, so that's what we were asking the question in this paper. And as so often happens in science, you start out answering one question and actually you end up answering something different. But we did manage to answer that first question, and to our surprise, the second tumour, DFT2, still expressed MHC class 1. So we're like, how is it possibly moving between individuals? And I don't think in this paper that we've actually been able to conclude this 100%, but what we've proposed based on our results is that actually the tumour and the host animals that are within that population actually share a lot of their MHC class 1 molecules. So in the human population, you have so much what we call diversity, a genetic diversity in these molecules, that you can't do transplants. But we think that perhaps that this tumour has kind of stayed at the moment within its favourite animals that have very similar MHC molecules to what it has.
It's sort of showing us how DFT1 could have evolved in the first place. It could have started out a bit like this and then evolved to have this downregulation of the immune response. This one is now doing exactly the same thing.
Yes, and if that's right, we're really concerned about how quickly the new tumour could spread, because if the fact that it has expressed MHC class 1 has kind of held it back or kept it in a constrained population, if it loses MHC class 1, presumably those constraints are off and it could really spread and transmit much more easily. And from what we know at the moment, it seems as if it is just as aggressive in terms of killing the animals as DFT1. And unfortunately, though, we have far fewer devils. They have less genetic diversity than they did 20 years ago before they got any of these diseases. So we have a much more vulnerable species.
And looking on the bright side, does what you've discovered shed any light on how we might be able to stop this thing?
Yeah, that's kind of the million-dollar question, isn't it? And I wish I had a better answer to that. One thing that my lab is working on is trying to design better vaccine strategies against this. And, that is a pretty tall order. But we have had some success against DFT1 that still needs more work, but does look as if it's promising. And I think at the moment, though, it's really getting a better understanding of these tumours, because if you think about it, if we didn't even know know that these tumours were losing MHC class 1, we might be quite complacent that this tumour was just going to stay confined to these animals. So really understanding, and I think that's a general lesson, isn't it? The more we understand about how diseases and pathogens work, then, the more chance we have of being able to come up with ways of preventing them spreading further.
Well, as they say, the devil is in the detail. Hannah Siddle there. You're listening to the eLife podcast with me, Chris Smith. Still to come, if your lab is headed by a woman, when it comes to getting your paper published, is that a good thing? Or might it be a bad thing? We'll find out. Before that, though, April the 19th, 1943 is a very special day for some, and it's commemorated as Bicycle Day. It's in fact the day that Albert Hofmann, a chemist working in the Sandoz pharmaceutical laboratories in Basel, took a dose of the LSD, lysergic acid diethylamide, that he developed in his laboratory. As he rode his bike home after swallowing the chemical, strange experiences began to unfold. Since then, we've learned a lot more about how this chemical affects the brain, and now, thanks to a recent study by Katrin Preller at the University of Zurich, appropriately enough also Hofmann's own alma mater, we now know which chemical receptors seem to be responsible for many of the effects of LSD in the human brain.
We had healthy participants in the MRI scanner, and they underwent 3 drug conditions. The first condition is just a placebo condition. The second condition was participants received 100 microgram of LSD. And the third drug condition was a condition where participants received catanserin, which blocks a very specific receptor in the brain, which is called the serotonin 2A receptor. And after that, we gave them the LSD to find out which effects that LSD induce are dependent on this particular receptor.
And who were the participants? What can you tell us about them in terms of what you knew about them when they embarked on the study?
Participants were 20 to 40 years old. They were all healthy. Most of them were students or interested people.
And had any of them used LSD recreationally, or did you not ask that?
Oh, yes, we did ask them. About half of them didn't have any prior experience. The other half did have prior experience, but none of them were regular users of psychedelics or any other substances.
And what sorts of experiences did the participants say they got when they took the drug?
First of all, what people notice is a visual alteration. They see the floor moving, they see colors changing, things might appear brighter. People feel some kind of disembodiment. Boundaries between yourself and the environment tend to dissolve, as well as feeling more connected to other people and to your environment. So people tend to call this alterations in self-experience.
And when you then put them in the brain scanner, do any of the changes you see explain the experiences that the people describe?
Absolutely. What we see is that there are certain areas in the brain which are dedicated to our sensory and sensory motor experiences. Those areas are connected more strongly with each other, so they communicate more. And there are other areas in the brain which are dedicated to integrating these experiences and making decisions, planning, and associations. And those brain areas, they are collected less under the influence of LSD. And if you think about what LSD does, I think that makes a lot of sense. So sensory awareness is enhanced, but it can maybe not be integrated as well, which then leads to hallucinations and differences in self-experience.
What about when you gave them the catanserin, the blocker for the serotonin receptors that you think might interrupt the LSD signal? Did that change their experience?
Absolutely. And this was actually quite surprising for us because LSD stimulates quite a lot of receptors, not only this particular one that we were blocking. And from animal studies, we knew that more of the receptors that LSD stimulates are involved in the subjective effects. However, when we blocked this particular receptor using catanserin, all the effects basically went away. They got normalized to a placebo level. So participants weren't able to distinguish between LSD plus ketanserin and the placebo condition. That means that the serotonin 2A receptor is crucial for the effects of LSD. And also when we looked at our fMRI data, all the effects that LSD was having on brain connectivity were basically gone. There was no difference anymore between the ketanserin plus LSD and the placebo condition.
Now, given what you've learned from this study, what are the implications of this, and might there even be therapeutic implications?
Yes, we know that LSD induces effects similar to some of the symptoms we see in schizophrenia, for example. So one of the ideas is that we could, for example, use the functional connectivity maps that we have seen here, compare them with functional connectivity maps from patients, and therefore find out who would be most likely to respond to a particular medication. The other one is, as we see here, we see that the alterations in the functional connectivity that lead to these loosening of self-boundaries, that might be something that is actually helpful in patients like depression who suffer from rumination and increased self-focus. And I think the alterations induced by LSD here might explain a mechanism why these psychedelics might work in a patient population.
Katrin Preller. Here at the eLife podcast, we try to create audio abstracts of some of the most important papers that are carried by the journal in recent weeks. But in each episode, we also try to shine the spotlight on some significant issues that are faced by practicing scientists. One of these is the rigour of peer review, where new science is scrutinised by authorities in the field before it's accepted for mainstream publication. But is this process done fairly? In the spirit of transparency, eLife made available to one of its editors, Jennifer Raymond, details of every manuscript ever submitted to the journal since its launch. She told me what she and her colleagues discovered when they analysed the data.
Well, the fundamental question is whether journals, when they review scientific papers, are finding the best science, or whether there are other factors, biases, that could be influencing the review process that journals use to decide which research to publish.
Now, when you say finding the best science, of course, journals don't go looking for publications. It's the other way around, isn't it?
Correct. The process is authors submit a manuscript that they would like to have published in the journal, and then the editors first make a preliminary decision. Is this work interesting enough or potentially interesting enough to send out to members of the scientific community for peer review? And typically 3 reviewers will write comments about the paper, send it back to the editors, and a decision is rendered.
Now, that sounds fair enough. And in fact, peer review is held up as the gold standard in how we should do things. So what's your beef with that?
The problem is that scientists are people, and we know that people are influenced by all kinds of cognitive biases.
So can I paraphrase by saying then, I get your paper and I decide, for whatever reason best known to me, I don't like you, so I'm actually going to can your paper. I'm going to say it's really bad, it's got lots of methodological flaws or whatever, and I'm going to say, let's kill it, let's not publish it.
Sure, that can happen. What we found in this study is more that people tend to give kind of an advantage to people who share certain characteristics with themselves.
Gosh.
And those characteristics include gender and nationality.
That's quite a claim. So how did you actually do the study? Let's look at the method first, and then we'll actually look at the results. So talk us through how you approached this.
First of all, we were very excited to get access to these data, every manuscript ever submitted to the journal eLife since its inception. First of all, we did what has been done a number of times, look at the differences in success rates of authors with different demographic characteristics. Women versus men, people from the US, UK, Germany, China, Japan. So we asked, what are the differences in success rates? But there can be differences in success rates that are not reflecting bias, because it may be that papers coming from certain groups are just of better quality, either because they've had certain advantages like access to research resources, or time or encouragement or other kinds of things. So seeing differences in success rates doesn't mean there's bias. And conversely, equal success rates doesn't mean there's no bias, because it could be that one group was actually better but had the equal success rate. So to try to get beyond that, we looked at how successful men were when reviewed by all male reviewers versus reviewers of mixed gender, and how successful they were when reviewed by reviewers from the US versus other countries.
And just to be clear, when you say success, that's a paper gets sent to the journal, it passes muster and ends up in print.
That's right.
And what does that show? When you feed these data through your sieve, what trends come out?
What we found is that the decision that the reviewers made about whether a paper should be published was not just influenced by the contents of the paper, but it was influenced by the demographics, by the gender and by the nationality of the authors, and by the gender and nationality of the reviewers. Male reviewers were more likely to accept a paper from a male author. People from a given country were more likely to accept a paper from somebody from their own country. So we call this homophily.
Was it just that one-way street? Was it just men favouring men, or did women tend to give more benefit of the doubt to other women authors?
There was a suggestion that it may be the case that women also give the advantage to women, but there were not enough cases of papers from women being reviewed by all-female reviewer panels because women are highly underrepresented at every stage of science. And in fact, we found that they were underrepresented as reviewers too. And when I say underrepresented, I don't just mean that not 50%. I mean compared to the number of women in the scientific community, they were invited to review papers less often than their male colleagues.
It's about 30%, isn't it? If you look at the workforce, about 1 in 3 scientists is female, 2 in 3 are male.
That's right.
And that's going to inevitably be reflected. If you think about, well, that's the pool of expertise we've got to rely on, it's probably going to be reflected in editorial boards and review panels and refereeship as well.
It's worse than that. We see that women are less represented as reviewers than they are as authors. So the author pool should be the same as the reviewer pool. And so we find that there's even less representation of women than we'd expect based on how many of them are submitting papers.
So you find that there's this homophily effect. Men look after men, women look after women. Does that apply regardless of who else is in the author list? So if you've got, say, a female first author, but a male senior author, does that skew things, or does it all ride on whether there's a woman's name there?
We did not find any effect of the gender of the first author. And it's really the senior author, kind of the head of the research laboratory, whose gender matters. And that makes sense because I think often the reviewers know the head of the lab, but might not know who the graduate student or postdoctoral fellow is who's doing the research.
Is one interpretation of that then that if I work for a female senior author and I send my paper off, I'm male, I nonetheless might face a publication bias against me owing to the fact they've got a boss who's a female?
That's true. It looks like papers coming from a given laboratory are influenced by the demographics of the senior author.
And these were statistically significant differences?
These were statistically significant differences, yes.
But obviously a statistician knows that could mean if you do a big enough study, which you did do a huge study here, it's thousands of papers, you can detect a very, very tiny difference. So how big was the difference in practical terms?
What we saw was overall manuscripts submitted by female scientists were accepted at a rate of 13.6%, whereas 15.4% of the manuscripts from male researchers were accepted. So, some people have said, well, that's tiny, that's 2%. Why are we worrying about that? But if you think of it another way, it means that the papers from the women are being accepted at only 88% the rate of the men. So there's kind of a 12% success gap there. And the effects are even bigger for some nationality effects. So, for example, papers from scientists submitting from China had a success rate of only 4.9% compared to 22.3% for researchers from the US.
So, should peer review go double-blind? What do you think? Jennifer Raymond there. Her manuscript, if you want to take a look at the complete study, is currently available on bioRxiv. Well, there we must leave it for this episode. You've been listening to the eLife podcast. Previous editions and full text transcripts for which, as well as the details on how you can subscribe, are available on the Naked Scientist website. That's at nakedscientist.com/eLife. This eLife podcast is produced by The Naked Scientists, and we also publish a weekly science programme covering the latest leading science stories. You can get The Naked Scientists podcast via your favourite podcasting app or from our website, that's at nakedscientists.com. I, meanwhile, are going to be back with another look inside eLife next month when we're going to discuss flying squirrels and a fly's taste for salt. Until then, thank you for listening, and from me, Chris Smith. Goodbye.
The eLife podcast. From eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.